My name is Hope Moore, and I have been assigned the KRAS protein with the mutation glycine AA12 is replaced by aspartic acid or cysteine, G12D or G12C, condition mutated in 10 to 30% of cancers. The normal function of this protein is a relay molecule involved in signal transduction. It functions as a molecular switch within the RAS/MAPK signaling pathway, transmitting extracellular growth signals. The KRAS protein is primarily located at the inner surface of the plasma membrane, where it needs to be functioned properly as a signaling molecule by interacting with other proteins involved in cell growth. It is also found in human cells such as immune and stem cells, and the KRAS protein is also found in animal cells. The protein becomes more active. Both G12D and G12C mutations stop KRAS from breaking down GCP into GDP, which is an important step to turn off its activity. Because of this, KRAS stays active all the time, constantly sending signals that drive cell growth. This is the nucleus, and the nucleus sends signals to make the cell divide, and this is the mutated when it sends stronger signals in the nucleus and the cell divides uncontrollably, which causes cancers. The original amino acid glycine has the simplest side chain of all amino acids, a single hydrogen atom, which is also the main functional group present in the original AA side chain. It is hydrophobic because its side chains consist of a single hydrogen atom, which does not form bonds or interact strongly with water. The charge is neutral. I would characterize the size compared to the other 19 standard AAs as the smallest amino acid, as its side chains is just a single hydrogen atom. The small size makes it unique and allows it to fit into tight spaces within protein structures. Glycine's small size gives it flexibility in protein structures, but it does not participate in many specific interactions. Its side chain can only form van der Waals forces, which are weak, non-specific interactions. The main functional groups present in the mutated side chain, the G12D mutation in KRAS replaces glycine with aspartic acid, which has a carboxyl group in its side chain. It is polar due to the carboxyl group, which can form hydrogen bonds with water or other polar molecules. The carboxyl group of aspartic acid loses a protein, making the side chain negatively charged. The side chain of aspartic acid is larger compared to glycine. It contains a carboxyl group, making it larger than many of the small amino acids. Compared to the original glycine, which has a small neutral side chain, aspartic acid can participate in ionic interactions due to its negative charge, as well as hydrogen bonding with water or other polar molecules. Glycine, being small and neutral, mainly participates in van der Waals interactions. The larger and negatively charged aspartic introduces more complex interactions that can significantly affect protein structure. The G12D mutation prevents KRAS from hydrolyzing GCP to GDP, keeping the protein in its active state. This leads to continuous activation of downstream signaling pathways, promoting uncontrolled growth. At the cellular level, this mutation drives the development of tumors. It can also contribute to various cancers, such as pancreatic and lung cancer, affecting overall health and survival. The limitations to this 3D model is that you aren't able to see the active and inactive states for the protein, and it also doesn't show how the cell division works. Another limitation is that it doesn't show how the cell travels and causes cancer to the body. Thank you guys for watching.